पुढे करायला नाही
तुम्ही आहे का स्टेजवर? तुम्ही आहे ना स्टेजवर?
A very good afternoon and warm welcome to today's speaker, Mr. Abhishek Dusane sir, all my colleagues and delegates who are attending this webinar. I, Dr. Ashwini Shelke, Assistant Professor, Department of Pharmaceutical Chemistry at Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nasik, welcome you all to third session of one-day national level webinar entitled Innovative Horizons in Pharmaceutical Sciences, organized by Department of Pharmaceutical Chemistry under aegis of Internal Quality Assurance Cell (IQAC). It's my great privilege to introduce you all to our third session speaker, Mr. Abhishek Dusane. Sir, we are having with us. Before going to start with session, I would like to introduce him. Sir is Senior Research Assistant at Sai Life Sciences Ltd. Sir has completed his Masters in Pharmaceutical Biotechnology from Pune College of Pharmacy, then Bachelor in Pharmacy from Sir Dr. M.S. Gosai College of Pharmaceutical Education Research and Nasi. Sir is having technical research skills in molecular biology, cell biology, electrophoresis, binding and kinetic studies, structural biology, computational biology. Looking at his work experience, currently he is working as a senior research assistant at Sai Life Sciences Ltd, Hyderabad, India. He worked as a senior executive in Sun Pharma Advanced Research Company, Vadodara, India. Then he also got junior research fellowship at International Center for Genetic Engineering and Biotechnology, New Delhi. Now I request to Mr. Abhishek Dusane sir to deliver his talk on topic drug delivery and development, current trends in pharmaceutical industry. Over to you, sir. Thank you.
Okay. Firstly, thank you very much, ma'am, for the brief introduction of mine. And also, thank you very much for giving me this opportunity to present my overall experience and share some insights into drug discovery and development. So, is my presentation visible, ma'am? Could you please confirm?
Yes, sir, it is visible.
Okay. So, starting with my today's topic, as you know, my title is Drug Discovery and Development, Current Trends in Pharmaceutical Industry. So, this would be some of the key points that I hope the presentation would walk you through. So, it's about drug discovery and development as to this might explain you as to how the overall drug discovery and development process is carried through overall, and what are some of the industrial aspects that we really look into or that we take care during drug discovery and development. Second, I would definitely like to point on small molecule versus biologics, because though there are many therapeutics available, but these are something which are currently mostly in the use, and these are going to be in future also. And in line with that, I would also put some highlight on what are some emerging therapeutic approaches that some of the industries and some good accommodations are also looking into. And so going ahead. So, first of all, as I said, it would be with drug discovery and development. So, I would like to start with this image where, it nicely fragments discovery and development into two different stages. So, any drug discovery, as we all know, would start with target discovery, because target discovery is one of the very very crucial aspect, because um, you know whole drug discovery process goes and reaches through most of the clinical trials, but hundred and thousands of molecule goes into, but most of the drug candidates fail into clinical trials because of n number of reasons, and one of it is good target discovery. If it would have been a good target, it is surely going to be a promising one. So, drug discovery meaning we do find target. Target in the sense in any given disease or its pathogenesis, there is some sort of drug target or biological target that we refer to uh, would be responsible for overexpression or down regulating some of cycles, and which would lead to the disease pathogenesis. So uh, this also entails further two different aspects, that is target identification and next is validation. So just let me so yeah, target identification and target validation. So once this part is done, we move ahead with hit generation and lead identification. These two are nothing but to find out a promising therapeutic molecule which would exhibit its pharmacological activity against that target, and down the line it would help to reduce the disease pathogenesis or completely reduce it to much lower levels. And going further, so once we have got the lead in our hands, we proceed for optimization. So optimization might roughly include what are the structural aspects that could be changed to get a more better activity. Further, it would also entail its profiling of its ADME. As we all know, how does it absorb, how it's distributing into our body, how it's metabolizing, how it's excreting. So this is much on the later part, which after optimization further goes into animal models where tox studies are done, ex vivo studies are done. So these are some crucial aspects that we do with when we have a lead in our hand. And after this so-called preclinical development, we go ahead with animal models and then clinical trials, which we all know goes with different phases. And once this is done, further regulatory bodies come into play, wherein they screen and monitor all the aspects, they check everything right from start to end, and basis on that and basis of regulation, they give some approval. And after which then the drug or the lead that we have got gets into market and is called as a drug. So this is a very rough explanation of what drug discovery and development means. And moving further, so as I mentioned, target identification is very important. And targets are nothing but mostly these days they are proteins. They might be nucleic acid also. And the table or uh, the image provided would give us the idea about what are different types of targets. Though there is a very detailed explanation to this, but something of highlighting that target might include your receptors, it might be your enzyme, it might be transporter proteins which is situated on your membrane and would be playing active role in transportation of various carriers or various drugs, various other uh, cellular factors maybe. And then it also includes nucleic acid. So then, how moving ahead? What are the different strategies that we reach to a target, and how do we identify it? So, uh, like there are molecular, there are system, there are computational, there are experimental. So uh, these are most commonly followed strategies into drug discovery, by which we reach to a target. So molecular might include clinical samples, that is clinical samples that we have from patients. Like there are say, some of clinical research organizations who are working on it, who can provide clinical samples, and those samples are then processed to they are analyzed by various modern biological tools to explore what all the proteins or what are the different factors that are overexpressed, that are downregulated. So in down the line, it would give us idea about what is something that is most prominent in this disease, and that would probably be a target. And that's how we learn with the things. So moving ahead with experimental. So experimental has genomics and proteomics. The reason why I am talking about genomics and proteomics is because uh, during these few years, this has been potentially emerging fields where they are actually dissecting what are the genetic factors, what are the proteins. They study at very deeper level, at RNA level, at protein level, as to what all things are uh, dominantly playing role in that disease, and making it to the pathogenesis. Also, uh, about target validation. So again, target validation is a process of dissecting the potential role of target in that disease. So once we have got any target, then it is also important that we do validate it. So for the validation part, uh, there are different aspects. So like protein modulation, mRNA modulation, cells and animals model. So protein modulation is also one of the thing, but it is not much of uh, it has some limitation, and hence uh, there are several strategies which in industries are studied or applied. So like once we have a target, we know that this is XYZ target in this disease. So we start knocking it out. So and study the if the same disease is happening or not in its absence or in its presence. So this is how we do the target validation. So there are some checkpoints that needs to be ensured. If these criterias are met, then we can consider that yes, target validation is uh, on a good level, and we can proceed ahead with this target. So because so once we start with this target, further there are going to be several of processes which would be going on with this target. And this would actually help us to analyze and to actually pinpoint in a very right way that yes, this is the target that actually is playing role. So what are the important criteria? So hence, so these are the important criteria that a target should probably met in order to be successful. So for any drug to act, uh, it should be very selective and specific, because once it's selective, there would be less chances that it would go and act to some off targets. So highly selective nature is one of the important parameters in that tangent. So three-dimensional structure, yes, it's always good to have its three-dimensional structure, and if not, yes, there are several techniques through which we can have a three-dimensional structure of that target, and go ahead with the process. So yes, then coming to, I personally believe that this is a very important point in so far when considering in vitro drug discovery, because basis on several enzymatic or cellular assays, it's only then we are gonna validate this target on experimental front. So I believe that it should be suitable for much of your biochemical and cellular assays, which would enable for high throughput screening, because there are n number of compounds, thousands of compounds which are going to be screened against this specific target. So the more it's favorable, more it has a good impact, and more chances of success would die down there. So yeah. So moving further, as I mentioned in my earlier slide, so this is for the drug target validation, identification. So once this is done, we move ahead to drug designing, which further goes up to lead optimization. So this is so I have tried to put in this image, which would give us a very basic idea of what to drug design look like, what are the starting points which we can start with. So basically, human genome sequencing, with this and with the available structure, most we can go with start with homology modeling, because for any drug to study its parameter, it's very crucial to have its three-dimensional structure. Hence homology modeling is one of the strong parameter by which we can start. And yes, again, protein extraction and purification, this also holds quite a good importance, because as I mentioned, not only drug targets, but also most of the protein drugs or therapeutics that we have in pipeline, or we have in market, are mostly protein. So this is how the first thing goes, that okay, you would do homology modeling, then you would go for if not, then we would like to have a 3D structure of that, because only when we have 3D structure we would be able to proceed further, because three-dimensional structure is not only about how the protein looks, but it also has a key components or key information which gives you idea about ligand binding pocket or drug binding pocket, because drug would not certainly bind to any region of that protein on any given surface. It has a very specific region where it would bind, and it would then stimulate or exhibit its activity, given therapeutic activity. So crystallography and NMR are most traditional methods which were used for studying the crystal structures. And it is not only with proteins, but it is also with protein and ligand. So ligand might be your natural substrate. So let's say there is like in enzymology, we study that for any protein or enzyme to act, we need a substrate. So only when substrate acts onto it, the protein would activate, and then down the line the activity would go on further. So this is so as I mentioned, it might be natural substrate binding site, it might be ligand binding site. So to counter on this part, and to have a very detailed insight into this, we need this crystallography techniques by which we can predict the structure. And certainly, as also mentioned by Dr. Mohd Usman, in silico techniques or in silico drug discovery has been playing, and is playing a very crucial role in compound screening, or because drug discovery is very much long going process, and it's very costly. So to reduce this time, to improve on efficacy, to improve on precision, in silico methods which are AI driven are certainly helping at its best to get the lead molecule. So now you really don't need to screen thousands of compound. Rather, you would need to filter only one fourth or one third of those compounds, uh, because the others would be filtered out. So based on that, we would have then a compound library which has to be then screened in experimental models. Then it would be your cell based, it might be enzyme based assays, and after that we would basis on that result we would reach to the lead compound. So uh, this is a very crisp or a very short, I would say, explanation of. So databases and softwares are two again important thing that we need, because that because these are AI generated things. So data is at paramount level. So for protein, we have protein data bank Uniprot, where we can uh, where we can actually get to explore the 3D structures, its sequences, its alignment, its amino acid structures, uh, not only with protein but also with some ligand which some scientific studies has tried and proven and to put front into scientific community. So again, for compounds, zinc is the most used. Enamin, Medicram Express. So these are the sources where you can actually have libraries of compounds. Uh, you can purchase them. You can use them in your studies. So this is about compounds. So coming back to software, so this information together would be applied into certain softwares where you can read some data and get out of some conclusion to it. So again, these are ChemDraw and Autodoc are very basic software that most of our medicinal chemists use for drawing, for predicting the structure. And Schrodinger Maestro is again one of the powerful technique which would not only get you done with docking but also with pharmacophore modeling, studying molecular dynamics, and henceforth. So, as I mentioned, homology modeling is a powerful computational technique which would help you to know about the 3D structure of protein. So, as we are talking about drug discovery, I believe certainly there are many out of which I have just put in two, uh, but these are uh, very common that we might encounter. So with that, it not only gives us the structure, but it would also give us binding site, because you can compare that with some of the previous structures and compare some sequences, because binding is about sequence, because a certain part of amino acid sequence would be responsible for binding of any XYZ. So if same would be there in your current or a new protein target, it would be good to have. So yes, so recombinant protein overexpression and purification. I would be emphasizing more on this part because, as previously mentioned, proteins are not only about drug targets, but it is also about therapeutic applications. So hence, recombinant protein overexpression or recombinant DNA technology or protein technology has emerged a lot and has helped a lot into this. So it starts with these procedures, or I would say these are three basic parameters or steps that we go through. First of all, hardcore molecular biologists who have a very core idea about RNA. DNA, how the protein is uh, like how protein is synthesized from its RNA. So the one who would have idea would go with cloning with plasmid or vector design, because vector is something or gene of interest which might be responsible for expression of that particular protein. So it's about sequence to protein, and there are two different types of vectors which we commonly get to see. One is cloning vector, one is expression vectors. So in protein expression, we are dealing with expression vectors, because these would be the one which when transformed in or transfected into a cell, it would initiate the expression of the protein which we are really interested into. About cloning, again, cloning is the vector where you can just incorporate your gene, keep it, and use it for several other studies in molecular biology. So again, Snapgene is one of the most commonly used software where you can design where you can put several parameters like antibiotic resistance, gene of your interest, and restriction sites, and those all things you can put into vector designing. So for overexpression part, it is done in bacterial, it is done in mammalian cells as well. So, but at majority, I would say bacterial is mostly used, because it is cost effective also, and most of your protein you can get expressed into bacteria. And the another advantage that you would have with this bacterial systems is that you can get larger amount of uh, proteins if wanted, or it is sufficient for you to have your most of the studies done. So bacterial is mostly studied and also used system. So again, nowadays mammalian cells, which might be insect, that is SF9 cells or yeast. These are also used as an expression system. So because uh, limitations to some of the bacterial system is advantage to mammalian system, because there are certain proteins which are of high molecular weight, uh, of greater kilodaltons, and it is very important for protein to get fold properly in its native form, to be in its native form. And hence for that reason, insect cell lines or yeast are choice which are made by scientists for overexpression of protein. So once the protein is overexpressed now, you need to purify it. So purification goes with this is, I guess, I would like to mention it as two-step chromatography techniques. So first is affinity, second is size exclusion chromatography. So down the line in this PPT, we would get to see more or get to learn more about overexpression and purification part. So here I have just tried to capture something with a bacterial expression system. So as I mentioned, this is your expression vector. As I mentioned, this is your gene of interest. So you would transform this into a competent E. coli cells. So you cannot transform into normal E. coli cells. You need to make it competent so that bacteria can actually have uptake of your gene of interest, and then it would further express into its system. So, so transformation is used when you are dealing with E. coli, and transfection is something when transfection is another terminology which I have not mentioned here, but it certainly deals with or has relation with when you say it's with your mammalian cells or insect cell lines or yeast cell lines or whatever you call. So, basis on that. Transformation is done into competent cells. you have to we usually give some time depending on antibiotic that we have used. So 12 hours, 24 hours, some also take 36 hours for bacterial colonies to grow. And once these colonies are grown, so yeah, just to add on to this part, we also keep control plates, whether if it's something else that has grown up on your bacterial plate. So, but ideally, only the bacteria which has resistance for the antibiotic that you have given would only grow in your media. So this is then taken to culturing part, where you would take this colony, you would incorporate into media where E. coli would have ample of time and space to grow into its system, and you would have thousands of copies of bacteria expressed, which would potentially have your protein, and then it would be harvested further. So in this, selection of media is one of the important part that comes to play. Media, you can use normal media like LB media, because there are some common strains which would easily uh, be grown into your LB agar media, sorry, LB media. Also, you have some advanced media where you have some components which would actually or potentially increase the bacterial growth. So you have to incubate this for dedicated hours. So usually this is measured in terms of OD, that is OD 600. So in meantime, in because bacteria multiplies in 20 minutes, so one cycle is completed in 20 minutes. So you have to get along with OD 600. So OD of zero point 0.6 to zero point 0.8 is usually considered as best OD that you can have. Once there is sufficient amount of bacterial growth that you have, you go with induction. So induction with IPTG, meaning which would actually start expression. Till this phase, we have generated a lot of bacteria, which would later which would later go for your protein synthesis. So for protein synthesis, to start the protein synthesis, we have to give IPTG induction. So IPTG induction, after IPTG induction, the bacteria actually starts to make the protein, which after certain hours is then completed. So once that's completed, you go with harvesting it. Harvesting meaning uh, the media, we decant the media, and the bacterial pellet which now has our protein expressed is then harvested and then stored in minus 80 degrees Celsius or minus 60 degrees Celsius for further use. So once we have a pellet, because now we have to get that protein out of this bacterial pellet. So we have to lyse these cells. So there are again another plenty of methods which are used for lysis. So after lysis, we would get cell lysate, basis on which we could go further with chromatography techniques. So potentially there are two, like as I mentioned, affinity chromatography and size exclusion chromatography. These are mostly used for most of the proteins and antibodies that that we have been working with. So just to give idea, so this is how the system looks like, the modern system looks like. So this is from SciTi. We have so advantage using these systems is that you can quantify the protein, how much protein you have diluted. It would give you idea of quality also, whether you would get a sensogram displayed on this desktop, where you can get single to multiple chromatograms. So basis on that, you can decide how pure is your protein, because multiple peaks meaning it might be contaminated, or may be some modifications with the proteins that you are dealing with. So, as I said, this is the chromatography that you do, and I'll explain this again in later slide in more detailed manner. So, once you elute the protein, you characterize, or you separate them using SDS page gel electrophoresis. So, as I was mentioning over here, now I would get into some details of this part, that is affinity chromatography. So, affinity chromatography is nothing but like the protein that you have been expressing, or when you were designing, you have certainly put some tag into that protein, which would in later stage would uh, help you to elute the protein. So these are the resins that we have. It might be uh, it might be of nickel NT, it might be of amylose, it might be on glutathione resin. It solely depends on what type of tag you have in your protein, because so the cell lysis, or the when you get the cell lysate, you have to pass through the resins. So as expected, if let's say if your protein has his tag, it would certainly have affinity for nickel NTA resins, and it would bind to it, and another remaining fraction would pass out through the column. So now your resins are bound with your protein of interest. So now we have to now we have to elute this protein. So or we have to take this pure fraction of protein. So before that, we have to give lot of washes in the sense, because there are multiple things which are unwanted and origin or sourced from cell lysate that we have previously put. So for elution, like for nickel NTA, we use imidazole. So if your protein had MBP tag, it would have been purified by amylose resin, for which maltose would have been elution buffer, and in this way you would get your purified uh, protein at the end. So this is a first step. So before proceeding to size exclusion chromatography, you would still have a certain amount of impurities present in your protein sample, which you have eluted. So this is because there are several endogenous protein which also has his tag, or some tag that would be binding to your resin also. So even that would come into your elute. So the reason why we proceed for size exclusion chromatography is that for most of the experimental studies that we do, we need to have protein more than 90 to 95% pure. So we further proceed for another chromatography, which is size exclusion chromatography. So here also you have resins, but it functions in a different way. So when you pass your protein, now it has molecules or proteins of various sizes. So one is your protein, and another is your or another few maybe your contaminants that which you don't want. So this is how that bead is designed, or this is how it looks like. So the larger proteins, or the proteins of larger size, would certainly be eluted in initial time or initial phases, and the small molecules, it would eluted slowly. So in that way you would have your contaminants separated, and you would get much pure fraction of your protein, like as this red color band is your protein of interest, which you need in purified form. So just to compare, I have put these two images, like how the nickel NTA chromatography looks like and how does SEC looks like. So there is certainly some difference into this. So you have got more cleaner protein, as you can see in this lane one, which is of your interest in one and both. So so once this you have got your elute, now you there is a reason for running SDS page. So SDS page is nothing but a technique based on electrophoresis, which would separate proteins based on their molecular weight. So so here, every protein has some sort of electrical charge. So in a way, we induce a method wherein, based on its charge, we separate the molecules. So ultimately, it would give idea or confirmatory idea about your protein, because your protein is, let's say, of 50 kDa. So it would be at 50 kDa you can find your protein, and you can confirm that yes, our protein is also eluted into this. This might also be used for some another purposes, like in some of the ex vivo studies, this protein is removed from this gel matrix and studied further if required. But that is on to later parts. So this is the general pictorial diagram of how we do gel electrophoresis and how it's carried. So any protein sample that we have uh, initially needs to be denatured or needs to be linearized, because only when it's linearized we would get to see bands like this. So in a sample we would add reducing dye, which would have potentially have bromophenol blue. Bromophenol blue is nothing but a dye which helps you to track about how the gel is running and to what amount gel has ran. Komasu blue is a dye that actually binds to your protein, due to which you can visualize the protein bands in SDS page or gel electrophoresis. So the sample initially is heated at 95 degrees Celsius for 5 to 10 minutes. It is then spent. So after this, then you are expected that with reducing agents like DTT, like beta mercaptoethanol, these are the agents which break the disulfide bond and make this circular protein into the linear ones, which is a crucial requirement for your SDS page. So with accompanied with heating, this certainly gets you to the gel electrophoresis, and then you get to see a picture like this. So again, I would like to put some highlight on crystallography studies. So now, once you have your protein purified and you know that it's more than 90 to 95 percent of purity, one would certainly like to have a structure of the protein. The more the purer, more is the refined structure. So this is how we proceed. Uh, this is a representative slide which gives us idea about how the crystallography studies are done. So when you have your protein in hand, with certain concentration, you move ahead for to make crystals like this. So the first step is that you want crystals like this, which can be put into your diffraction machine, which would be diffracted, and basis on which then you would get this protein like this protein structure. Though it involves a lot of deeper steps in between, but this is a very general overview of how the crystallography studies are conducted. So initially, as you can see, this is a crystallographic robot. We also call it as mosquito. So there are multiple uh, uh, spaces where you can put your plates, which has your protein, which has your crystallization reagents like PEG, polyethylene glycol, or some other salts which are crucial for crystallization. So once you do this, so this you can do in this two ways. So this is single drop technique, one is hanging drop technique. But so in sitting drop or single drop, you can see that this is how the protein sample is kept. Here is the reservoir solution like PEG or another salts, which would actually help to crystallization. So, what is the normal thing that is happening into crystallization? How is protein crystal form? So, when you get the protein, it is into the buffer. Buffer has some aqueous reagents, some salts, some another components. So ultimately, what these approaches are doing is it would uh, uh, it would get removing your water or solvent molecules, leaving behind the concentrated protein, which is then good and ready for crystallization. So with this phenomena, protein crystal is formed, which is then again looped into crystallography machine, and then we get to see a structure like this at the end of the day. So starting with the drug screening part. So as mentioned by Dr. Modusman, he has already given us a very nice insights into in silico screening. So I would just be giving overview, like these are the strategies that are considered when we think of in silico drug discovery. So one is structure based drug design, one is ligand based drug design, de novo library. So all has their different meanings. So let's say for structure based drug design, when you have protein structure known, when you have ligand structure known, so that is a structure based drug design. So this would give us an idea about what basis on structure you would be doing drug designing. So it would help you to get like what are the functional groups, or what are the moieties of that particular drug compound that is actually binding to that structure, and to which part of structure it is binding. So uh, this is the overview of in silico screening. I won't be going deep into that because it has already been explained, and also due to some time constraints. So yeah, so again, once we have some compounds from this in silico screening that we know surely are good hits, then we go for high throughput screening. So for any high throughput screening, first thing we need to keep in place is assay, because it's on basis of assay that you would be determining or screening the compound. So asset development and optimization is very much a crucial parameter into high throughput screening. So asset development, it certainly means that what kind of model you would be screening your compound, whether it's enzyme based, whether it is cell based. So uh, enzyme based, when we would think of enzyme based, because there are certain targets, as mentioned previously, those are enzymes. So for that we can go with enzyme based assays. Cell based model is when you need to have some deeper idea about its regulation, about its upregulation, downregulation. So in that case we would be using cell based models. So, what are the optimization parameters? So, this is again one of the crucial segment when we think about asset development or its optimization. So, solvent or buffers that we would be using. Because it has certain salts, it has some another reagents like surfactant, which might be potentially affecting your drug screening, or it would be also helping. So to optimize solvent or buffers, it is uh, mandatory to uh, optimize, because let's say per se, for some example, there is some drug which has some solubility issues, or let's say there is a protein biomarker which you are studying as a drug target, so it does has solubility issues. So adding of some of the surfactants or increasing the salt concentration might increase its solubility and making it suitable for assays. So second is pH. Again, for the protein, pH is most important parameter to be considered. Temperature, incubation, and then plate formats. So temperature and incubation at what temperature till what time. So we need to place it at some different temperatures at different time points, and then we optimize that acid. And one particular thing we get down to, and that is further used for the as a parameter. So we have different plate formats. So again, we have to decide which plate format we have to use. About spectroscopy, uh, then some are color based, that is absorbance based. Some are fluorescence based. Some are luminescence based. So basis on that, you have to decide as to what technique you would be using and what plates you would be using. Because let's say for fluorescence microscopy, or uh, the assays where fluorescence is something that has to be measured, for that you need to use Blackwell plates. So in this way, various parameters are considered, and uh, move ahead with the further process. So again, once you have set of libraries, you need to test actually how toxic that particular compounds or set of compounds is for your drug, uh, sorry, for your protein. So for that we do toxicity assessment, and these are two commonly used assays, that is MTT and CTG. So CTG is mostly used assay these days in compared to MTT, because it directly in much greater sense, it gives you idea about cell viability, or how many number of cells are alive, and how much cell death is there. Because CTG directly measures your cell nucleus ATP. So, and hence CTG is nowadays preferred option over MTT if allowed. So second, so you have to study that drug, but in what sense you have to study? Whether it's about allosteric binding, is it competitive binding, or you have to see some inhibitory or effective activity. So allosteric binding, as we have studied in pharmacology, and we know that uh, there are some drug targets which has some allosteric binding sites. So we need to look for these all parameters also. So this mode is also something that we take into consideration. Again, for that this particular target, we need to have a tool compound or a reference compound, because this would help us to have a reference as a reference, and then basis on that we would judge the another compounds uh, which we are interested to screen. So, in high throughput screening, when we are talking about, initially there are thousands of compounds. Let's say there is library of 50,000 compounds or 70,000 compounds. So, it is practically not possible to have many concentrations at a certain point and then evaluate any IC50 or EC50. So, at instance, only three particular concentrations are taken, which gives us idea about what range needs to be selected, and then the filter ones would be then tried for multiple point DRC. So there are some of the technological platforms that are amazingly boosting high throughput screening, out of which like liquid handlers. So liquid handlers are nothing but it is for more accuracy and precision. And the another advantage is that you need to spend only a nanoliters of your samples, because during synthesis or synthesis of compound, or let's say any protein drug, we know that the costs involved are much, and the sample or compounds is very much precious to you, and it has volume limitation. So in that case, these are liquid handlers which within fraction of seconds would dispense any amount of this range that I have mentioned to your 384 well plate, or whichever plate you are using for. Again, the liquid dispensers. So liquid dispenser, initially we used to do with manual pipettes and everything, but now there are liquid dispensers which precisely would dispense the designated volume to your plates. And so this in a sense are to to reduce the time and cost, which are one of the crucial factors in any drug discovery program or process so far. So then, hit to lead is uh, uh, another major parameter. So once here in high throughput screening, you would have certain set of compounds that you know now are best in their class. So you need to find lead amongst this. So there are various assays though which are used. So hit to lead, to put it in more precise way, has your selectivity assays, has your IC50 and EC50 predictions, which would give you idea about a more and more precise and detailed idea about how effective your drug is for that given target. So thermal shift assays, as mentioned, these are again commonly used in both academics and in some startups or some large scale industries also. So you you screen it for thermal stability, meaning how sensitive is your protein for different ranges of temperature, and what is the suitable temperature. This would again include some of the advances in techniques like nano DSM, that is differential scanning fluorometry. So this would again additionally give you idea about size analysis, thermal stability, and also some another parameters. So this is one of the aspect. Another key aspect is protein drug interaction. This is also one of the uh, aspect which is of great importance, because here you would actually screen these into some of your functional and selectivity assays. So for protein drug interaction, when it's considered, we have fluorescence polarization assays, we have some advanced techniques like surface plasmon resonance, isothermal calorimetry, and also octet systems, which I have not mentioned here, but those are also used. So again, selectivity assays, like I mentioned previously, now once you have done 3-point DRC, you can go with multiple point DRC, by which you can calculate IC50 or EC50, inhibitory or effective concentration. So since I am currently engaged with SPR work, just I would quickly like to highlight on what this techniques is about, and what it gives us, what kind of information does it gives. So in it, it works on basis of a change in refractive index. So this is a gold coated uh, glass which have capacity to generate plasmons when a light source is embedded into this. So the idea is that you would immobilize your protein of interest into this to study one-on-one interaction with your protein. So once the protein is immobilized, so this also has different chemistries like amine coupling, and it may be histidine. So depending on your tag, you would use the chemistry. So you would immobilize the protein, and with this fluidic system, you would then pass drug your samples which has your drug or compound in different concentrations. So you would pass and then see what is the binding and affinity parameters. So this is how it works. Like first it would associate, then it would reach the steady state, that is equilibrium. Then further, once the injection is stopped, it would dissociate and again reach to its baseline. So this Ka that I have mentioned is about speaks about association or association rate, or also we call it as on rate. Kd is again your off rate or dissociation rate, and when uh, we divide both of these, we get Kd. So Kd is nothing but it gives us how much amount of your ligand is bound when it's in equilibrated state. So this gives us highlights about how good is that compound binding to your protein, and how good is the affinity for that particular protein. So so further, there are several aspects to this which I mentioned in earlier, like in vivo studies, ex vivo studies. So which are collectively called as preclinical studies, are for the later part. But I wanted to highlight this, and due to time restriction and basis on my experience, I have tried to put in something more for discovery part and not development part. So along with this, now as we deal with drugs, so we also need to know that means we generally know that there are small molecules versus there are large molecules or biologics. So these are certain key parameters to it. So size-wise, when we speak, it's small. Biologics are large molecules. Uh, in terms of structure, small molecules are very uh, simple in structure, while biologics are very much complex in structure, because most of the biologics that we talk about are proteins, are enzymes, are antibodies. So these all are biologics are nothing but more like biomolecules which are of higher uh, molecular size and weight. So talking about stability, so uh, relatively stable. So biologics, so far when it's considered, so it has great concerns with storage and handling conditions, because they are sensitive to temperature, they are sensitive to some of the contaminants that might be encountered during various storage or handling. So the reason why I am willing to differentiate between small molecules and biologics is that both have their own advantages and disadvantages. But precisely and certainly, biologics are one of the emerging class of drugs. It might be antibodies, it might be vaccines, and great example we have seen in this last COVID-19, where vaccines have helped greatly, which again are biologics. But there are many developments into pipeline which are taking place, and which are making biologics as more suitable candidates for therapeutics. What are the emerging therapeutic strategies apart when we talk? Like dual drug approach. Dual drug approach is nothing but these days there are some protein, or let's say there is enzyme. So when an is natural substrate is acting or activating that protein, the protein further initiates its activity. Here there is a need that we inhibit the binding of natural substrate to the its binding pocket. Dual drug approach is where we use two different drugs which forms a good complex and then binds to that particular target. So one is active site, one is your natural or natural substrate binding site. So in that way, you would inhibit the further activity of protein. Again, targeted protein degradation. This is also one of the interesting approach coming up as a therapeutic option. It's nothing but it pairs the proteins that typically don't interact. Let's say the proteins which are undruggable. It would selectively go and it would perform its function of degradation. Examples are molecular glues and protacs. When I talk about molecular glues, the molecular glues are nothing but certain or a specific or a typical molecular structure which would bind two proteins together and which would ultimately lead to degradation. This is targeted protein degradation. Again, another one is antibody drug conjugates. Yes, this is again one of the upcoming ones, and much of the work is already done and is been carried on this antibody drug conjugates. So this typically has antibody that is covalently attached to your cytotoxic drug via chemical linker. So you would have antibody, you would have your cytotoxic drug, which is linked with chemical linker, and then it would help to potent killing or elimination of the cancer cells, which I would like to highlight over here. These strategies are becoming interesting ones and preferred ones because complexity of diseases nowadays are increasing. Let's say for uh, some of the diseases, mostly like cancer, because cancer is one of the diseases which has been studied for years. Many of oncology programs have are putting up new information, but still we are not able to reach to any specific therapy which would actually uh, control or completely eliminate the cancer without much of its side effects or adverse events. This was something that I wanted to present. So thank you for your time and attention, and welcome to any further questions.
नाशिका दिसतोय ना. आपलं आहे काय खबर? तुम्ही अशाने दिसता इथे दिसतच नाही. बोलायला स्टार्ट करून देतो. Thank you, sir. Delegates having questions, you can text your questions in chat box. Yes. If delegates are having any question, okay. As there are no questions, thank you, sir. Thank you for giving insights on different aspects of drug discovery and development in pharmaceutical industry. Sir has emphasized on strategies to target identification, homology modeling, recombinant protein over expressions. It was a very definitely informative and elaborating session related to the drug discovery and development, surely which is going to help the participants in their research work. Thank you, sir. Now I request you to please accept the certificate as a token of love. Thank you, sir, for taking time out of your busy schedule and joining us for today's session. Thank you, all
That's
participants, for making our webinar successful. Uh, Feedback link has been posted in a chat box. Participants can fill the feedback form. Okay. Thank you very much. Thank you, sir.
thank you, thank you.
Uh, Participants, please wait for 5 minutes. कॅमेरा ऑन आहे तरी कॅमेरा तो ऑन.
मग आपलं हे राहू द्या ना.
कॅमेरा ऑन राहू दे, दिसत नाही. Good afternoon to everyone who are virtually present for this webinar. Myself, Dr. Shilpa Hara, Associate Professor and Coordinator of this webinar on behalf of Principal and faculty members of our college, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, feel privileged and honored to pledge my gratitude to all those who contributed to make this webinar a success. It is said, learning is not attained by chance. It must be sought for with ardor and diligence. Webinar is one of the most convenient and popular way of gaining knowledge in the recent days. With this motto, we have continued the webinar series in the third consecutive year. In 2023-24, we had 7,000 beneficiaries. In 24-25, we carried out 7 webinars and we had almost 13,000 participants benefiting from this. So today's webinar, which was titled Innovative Horizons in Pharmaceutical Science, has reached an audience of 1,786 participants. Thank you, participants, for such an overwhelming response. So, I would offer my sincere thanks to our today's first speaker, Dr. Anindita Behra, Professor, MIT University, Kolkata, for her insights in the role of nanotechnology in brain drug delivery. Thank you very much, ma'am. I am grateful to Dr. Usman Siddiqui, Associate Professor, SVKM Institute of Pharmacy, Dhule, on making CAD, computer-aided drug design, so simple to understand and do. Thank you very much. A heartfelt thanks to our very alumni, Abhishek Dusane, for his insights on the drug discovery and development, and the related current trends which he discussed. Thank you, Abhishek. I am grateful to all the resource persons for sparing their valuable time with us from their busy schedule and enlightening us on the innovative horizons in pharmaceutical sciences. I am thankful to Dr. R.P. Deshpande, sir, President, Gokhale Education Society, for moral support and guidance. I also wish to express my gratitude to Dr. Dipti Deshpande, ma'am, Secretary, Gokhale Education Society, for her motivation and constant support. I would like to express my deep sense of appreciation to our mentor, Sri Shailesh Gosavi sir, Director, Establishment, Gokhale Education Society, for his unflinching faith and trust in us and his constant guidance and motivation. A special thanks to Dr. S.V. Amrutkar sir, principal of a college, for his enormous motivation and cooperation in organizing this webinar. I am thankful to Dr. P.L. Pingwe, vice principal and IQAC coordinator, and also the convener of this webinar, for providing immense support to make the webinar successful. I convey my thanks to the organizing committee members and my colleagues for working hard for the past few days to make this webinar a success. I especially mention a vote of gratitude to support staff, without whose cooperation and support today's success was not possible. Finally, I am thankful to all delegates for their active participation, and I hope the participants have benefited by attending this webinar. Those who could not attend it live, do go back on YouTube on IQAC channel of our college and watch the videos. I'm thankful for everyone who has made a contribution to this successful event. Thank you all. The feedback link has been posted. The feedback link has been posted on the YouTube channel in the live chat, and also it is being posted in the WhatsApp group. So I request everyone to fill the feedback form. You will be receiving your certificates within next working 10 days. And I invite you to join us again next month on the seminar. Thank you.